Hello friends, in the last few videos we discussed about enzymes involved in DNA replication. We discussed about Helicase enzyme, Primase enzyme, Topoisomerase enzyme and single strand binding proteins. You can refer my videos, the link is given in the description box. Today we are going to discuss about DNA polymerase enzymes. In this, we will discuss about general features of DNA polymerases. Then we will discuss about types of DNA polymerases in prokaryotes. Then we will discuss about types of DNA polymerases in eukaryotes. And at last, we will discuss about comparison of DNA polymerases in prokaryotes and in eukaryotes. So, stay tuned. Let us see general features of DNA polymerase enzymes. DNA polymerase in prokaryotes. It was first purified and characterized by Arthur Kornberg in E. coli. What are DNA polymerase? These are group of enzymes that catalyze the synthesis of DNA strand on template strand during DNA replication. So, these catalyze the synthesis of DNA molecules from nucleoside triphosphates. And these nucleoside triphosphates are molecular precursors of DNA. Now the functions of DNA polymerase. First, DNA replication. It is the main function. DNA polymerase duplicate or replicate DNA content of cell during cell division. Thus, genetic information is maintained and transferred from one generation to another. Second is repair. It maintains integrity of genome. How does DNA polymerase work? Main function of DNA polymerase is DNA replication. Existing DNA strand is read and two new strands are created, same as existing one, so that each daughter cell during the cell division receives similar DNA molecules. Chemical reaction that is catalyzed by DNA polymerase during replication is DNA N. N indicates N number of deoxynucleotide triphosphate plus 1 deoxynucleotide triphosphate. Now DNA is increased by 1 deoxynucleotide triphosphate represented as DNA N plus 1 plus inorganic phosphate. Direction of replication is 5' to 3' always. Why? Because DNA polymerase can add the nucleotide at the 3' end only, and one nucleotide is added at a time. So the directionality of the daughter strand, which is 5' to 3' is opposite to the direction of the template strand, which is 3' to 5' along which DNA polymerase moves. As a result, resultant double-stranded DNA is composed of antiparallel strands. So these two are the parental strand or the template strand. DNA polymerase moves in 3' to 5' direction, here and also here, and the new strand is synthesized in the 5' to 3' direction, this strand and also this strand, as a result of which the new DNA molecule that is obtained has antiparallel strand as the parental DNA molecule. One has 5' to 3' in this direction and the other has 5' to 3' in this direction. DNA polymerase can add nucleotides at 3' OH group but cannot initiate new strand, hence primer is needed. What are primer? These are segment of DNA, RNA or both. Generally it is RNA. Now the processivity of DNA polymerase. It is average nucleotides added by a DNA polymerase per association with the template strand. So, DNA polymerase associates with the template strand for the synthesis of new strand. And these add nucleotides one by one in order to synthesize new strand. So, number of nucleotides or the average nucleotides that DNA polymerase adds in one association is known as DNA polymerase processivity or processivity of DNA polymerase. Replication is highly accurate process, and if there is change in single nucleotide, it leads to mutation. There are two mechanisms by which DNA polymerases make sure that there are no errors. First is the geometry of active site. Only correct nucleotide base pair fit. So the geometry of the active site of DNA polymerase is such that it allows only the correct nucleotide base pair to fit. But still, incorrect nucleotide is added, but it is rare. It is added after 10 power 4 to 10 power 5 nucleotides. 3-5' exonuclease activity of the DNA polymerase, which is known as the proofreading, helps in the removal of any error if it is incorporated in the DNA. So in this proofreading activity, when incorrect base pair is recognized, DNA polymerase move backwards by one base pair. Incorrect base is excised. This is followed by reinsertion of correct base. This is known as proofreading. In this way, integrity of the original DNA strand is preserved and it is passed on to daughter cells. But still errors arise in the DNA, and this is due to replication errors and errors due to DNA damage. DNA is damaged by various factors. Among them, one of the most important is by the UV radiation. It is important to maintain the fidelity of the DNA during the replication, or the fidelity is very important in the DNA replication, because the mismatches in the DNA base pairing result in dysfunctional protein and can lead to cancer. So it is very important to detect the error that is incorporated in the DNA strand and how this error is detected. The shape and the interactions that accommodate Watson and Crick pair contribute in the detection of error. We have discussed that DNA has double helical structure, which was proposed by Watson and Crick, where adenine always pairs with thymine and guanine always pairs with cytosine. But if any wrong base is incorporated, then it can be detected and the error can be removed. So how it is detected and then the correction of the error is done at a mismatch, that is, if there is mismatch base pair, then there occurs loss of interaction between the base pair, and this loss of interaction triggers shift in balance from polymerase to exonuclease domain for the binding of the template primer. Now the exonuclease domain binds to the template primer in order to correct the error. DNA polymerase has polymerase and exonuclease domain, which we will discuss in detail in the next video, where we will see types of DNA polymerase, their structure and function. So if we see this figure, this is the template strand and this is the new strand which is synthesized by DNA polymerase. If we see here, template has cytosine strand and cytosine always pairs with guanine, but here adenine is added. So here incorrect base is added and there is mismatch, as a result of which these two base pairs do not interact with each other. This results in the shift from the polymerase domain to the exonuclease domain. Now it functions as 3-5' exonuclease. It moves backward by one nucleotide. Incorrect base, that is adenine, is removed and correct base guanine is added, and then again there is shift from exonuclease domain to polymerase domain and replication continues. This is known as proofreading. So what we can say about proofreading, that when wrong nucleotide is incorporated, it causes retardation in the DNA polymerization. And due to this delay, DNA polymerase is switched from polymerase site to exonuclease site, then the error is corrected and then DNA replication again continues. DNA polymerases are also involved in post-replication repair of DNA, which we will discuss in detail in separate video on repair mechanism of DNA. Now fourth is translesion synthesis. To avoid the harmful consequences of a stalled replication fork, certain specialized polymerases, example polymerase 4 and 5, traverse the damage. When there is damage or lesion in the template strand, then DNA polymerase 3 stops, extending the new strand, as a result of which replication fork is stalled. In that case, certain specialized polymerases, example polymerase 4 and 5, which are also known as SOS polymerase, travel past the damage and continues the extension of new strand. And this is called translesion DNA synthesis or TLS. This provides the cell additional time to repair the damage, and once the damage is repaired, replicase, in this case DNA polymerase 3, returns to complete genome duplication. Let us understand it in this way. When lesion is encountered in the DNA template during the replication, replication fork stalls or DNA polymerase 3 stop extending the new strand or replication of the DNA. DNA polymerase 3 then dissociates from the template strand and it is replaced by translesion DNA polymerase, example DNA polymerase 4 and DNA polymerase 5. These are known as SOS DNA polymerase. These extends the DNA synthesis across lesion or damage, that is, these traverse the damage or travel past the damage and continues to extend the DNA synthesis. In this way, cells get additional time to repair the damage. And once the damage is repaired, replicase, that is DNA polymerase 3, returns to complete duplication of DNA. Now, types of DNA polymerase in prokaryotes. There are 5 types of polymerases. First is DNA polymerase 1. These were first discovered, and it was discovered by Arthur Kornberg in 1958. It consists of single polypeptide chain. It is known as metalloenzymes as one atom of zinc is present per chain. It is most abundant. Greater than 95% of polymerase activity in E. coli is of DNA polymerase 1. These has poor processivity rate. These add 15 to 20 nucleotides per second. Now the activities of DNA polymerase 1. These have both polymerase as well as exonuclease activity. First is 5-3- polymerase activity for the new strand synthesis. This synthesizes the new strand that is complementary to the template strand. 3-5- exonuclease activity, that is removal of mismatched nucleotide or in nick translation. This also we have discussed in detail in previous video. Then 5-3- exonuclease activity, by which there is deletion of RNA primers from 5' end of complementary DNA strand. Hence, these are involved in the processing of Okazaki fragments and gap filling. Let us understand this with the help of the diagram. This is the leading strand, this is the lagging strand. So, DNA polymerase 1 removes the primer from the leading strand and as well as the lagging strand by the 5-3- exonuclease activity. Lagging strand is in the form of the Okazaki fragment. When the primer is removed, then there is gap between the Okazaki fragments. And DNA polymerase also helps in the filling of these gaps. Now, the structure of DNA polymerase. These resembles the right hand of human. And these has 3 regions or domains. First is palm region, which consists of beta pleated sheet. These are the catalytic active site. DNA binds to this palm region. and its primary function is processing the addition of deoxynucleotide triphosphate. Second is finger region or finger domain. Primary function is to bind the nucleotide triphosphate with the template base. Third is thumb region or thumb domain. It binds the recently synthesized DNA and maintains the correct position of primer and active site. Hence, plays a potential role in the processivity, translocation and positioning of DNA. This is the structure of DNA polymerase 1. This is thumb region of finger domain and palm region of palm domain. This is the template strand and this is the primer with 3-OH end. This is the incoming nucleotide. This template binds to the palm region of DNA polymerase 1. Second is DNA polymerase 2. It was discovered by Thomas Kornberg in 1970. It is made up of single polypeptide chain that consists of 7 subunits. It is known as the backup enzyme of polymerase 3. Activities, it shows 5-3- polymerase activity. That is, it adds nucleotide bases for the new strand synthesis. It is less efficient than polymerase 1. It also shows 3-5- exonuclease activity, that is removal of mismatch bases or nick translation. So during SOS induction, that is when there is mismatch bases present or there is nick, its cell presence jump from approximately 30 to 50 copies per cell to approximately 200 to 300 copies per cell because their activity increases or these are involved in the 3-5- exonuclease activity. Structure. The structure is quite unknown. Now the third type of DNA polymerase is DNA polymerase 3. It is primary holoenzyme. Holoenzyme is completely functional enzyme which consists of both enzyme and coenzyme. Its main function is DNA replication. Hence, it is known as the major replicative polymerase for both leading and lagging strand. It consists of 2 polypeptide chains and has 10 subunits. Now, the activities of DNA polymerase 3 enzyme, 5-3- polymerase activity. That is addition of nucleotide bases for the synthesis of a new DNA strand. So, it helps in the synthesis of the new DNA strand which is complementary to the template strand. 3-5- exonuclease activity. That is deletion of mismatched nucleotide bases or helps in nick translation. This we have already discussed in detail in previous video. You can refer it. Now let us see the structure of DNA polymerase 3 enzyme. It consists of 10 subunits which we discussed earlier. First is alpha subunit. It helps in DNA synthesis. Hence, it is known as the polymerase activity hub. Second is epsilon subunit, which helps in the 3-5- proofreading activity. Third is theta subunit. It acts as an accessory protein and it also participates in proofreading mechanism. Then tau subunit, it promotes dimerization of the core protein complex. Gamma subunit, then delta subunit, it is exonucleolytic proofreader. Then delta dash subunit, then xi subunit, then psi subunit, and at last is beta subunit. It acts as a clamp protein that holds DNA molecules and is responsible for processivity factor. So, if we talk about DNA polymerase 3 structure, it is made up of clamp loading complex, beta sliding clamp, and third is polymerase 3 core. So let us see them one by one. First is clamp loading complex. It is made up of gamma, delta, delta dash, xi and psi subunit. All acts as a clamp loading complex and helps beta clamp to bind with DNA. Second is beta sliding clamp. It is present as duplicate or dimer. Dimer of beta subunit forms a ring-shaped structure. It is involved in the processing of both leading and lagging strands. Third is polymerase 3 core or core domain. There are two core domains. Each of them are made up of alpha, epsilon and theta subunits. These are also known as catalytic core of DNA polymerase 3. It contains polymerase activity and 3-5- exonuclease activity. So this is the structure of DNA polymerase 3. This is core domain which consists of alpha, epsilon and theta subunit. This is beta clamp which consists of dimer of beta subunit. This is clamp loading complex which consists of gamma, delta, delta dash, psi and xi subunit. In addition to these three parts, tau subunit is also present. It is present as dimer. It is known as dimerization component. It causes two core polymerase molecules to link together at the fork or the replication fork, so that both leading and the lagging strand is synthesized or processing of both leading and lagging strand takes place. Now the next is DNA polymerase 4. It is part of Y family of DNA polymerase. It has no 3-5- exonuclease activity, or in other words proofreading activity. Hence, it is error prone DNA polymerase. It is expressed by DIN-B gene. This gene is switched on via SOS induction when DNA polymerase 3 is stalled at replication fork, and the production of polymerase 4 rises tenfold. Now the function of DNA polymerase 4. It interferes with the polymerase 3 processivity. A checkpoint is created, hence replication stops and provides time for DNA lesions to repair. Another function is translesion synthesis at stalled replication fork or TLS. This we have already discussed in previous video on general features of DNA polymerase. You can refer it. The link is given in the description box. Now the DNA polymerase 5, it is also part of Y family of DNA polymerase. Like DNA polymerase 4, it has no 3-5- exonuclease activity, hence it is also error prone. It can use translesion synthesis on lesions that block replication or miscoding lesions which modify bases and lead to wrong base pairing. It is highly mutagenic. Hence, it is used as a last resort in DNA repair mechanisms. Now, let us discuss DNA polymerase in eukaryotes. According to scientists, there are at least 15 different polymerases present in eukaryotes. Among them, 5 have been studied more abundantly. These are as follows. First is DNA polymerase alpha. It is member of family of B polymerases. It is unique or unusual enzyme. And activities which it shows is 5-3- DNA dependent DNA polymerase. Largest subunit has this activity. Then 5-3- DNA dependent RNA polymerase activity, which is also known as the primase activity. Smallest subunit has this activity. It is the only enzyme which have both activities. Hence, it is capable of self-prime DNA synthesis, that is, initiate a new strand. Or we can say that it synthesizes its own primer and then synthesizes new strand. That is, it itself is able to start the new strand synthesis. None of the other DNA polymerase enzymes are capable to initiate a new strand. But it lacks intrinsic 3-5- exonuclease activity, that is the proofreading activity. Hence, DNA that it synthesizes is inaccurate. It also lacks 5-3- exonuclease activity. It has low processivity. So, it is a poor candidate for major replicative polymerase, or we can say that these are not the major replicative polymerase. And the primary function that it performs is to make short RNA DNA primers during DNA replication, which is approximately 10 bases. This is followed by synthesis of 20 to 30 bases of DNA. Then this DNA polymerase alpha is replaced by an enzyme that results in extension of DNA strand. Here these are polymerase delta or polymerase epsilon. So this event in which DNA polymerase alpha is replaced by major replicative enzyme DNA polymerase delta or DNA polymerase epsilon is known as polymerase switching. Now the second is DNA polymerase delta. It is main enzyme for replication, or it is major replicative enzyme. It is highly processive in the presence of pCNA, proliferating cell nuclear antigen. So it is associated with pCNA, as a result of which it is highly processive. And this pCNA is equivalent to sliding clamp beta of polymerase III in prokaryotes, which we have already discussed in the previous video, types of DNA polymerases in prokaryotes. You can refer it. The link is given in the description box. Activities that it performs are 5-3- polymerase activity and intrinsic 3-5- exonuclease activity, and because of these activity it is main enzyme for replication or major replicative enzyme. Now the functions performed by DNA polymerase delta enzyme. It synthesizes the complementary strand. It uses the parental strand as the template and synthesizes the complementary strand. It displaces the primer or removes the primer. Also, it is involved in some types of repair synthesis. Now DNA polymerase epsilon enzyme. It is member of B polymerase family. It is main polymerase involved with DNA replication. Or like DNA polymerase delta, it is also major replicative enzyme. It is also processive enzyme as it is associated with pCNA, that is proliferating cell nuclear antigen, like DNA polymerase delta. So the activities that it shows is 5-3- polymerase activity and second is 3-5- exonuclease activity. It is involved in following functions. Involved in primer elongation in addition to polymerase delta. So we have seen that it is major polymerase enzyme like the polymerase delta. Hence, after initiation of synthesis by polymerase alpha, polymerase delta and polymerase epsilon synthesize opposite strands of DNA. Or in other words, if one strand is synthesized by polymerase delta, then the other antiparallel strand is synthesized by polymerase epsilon. It participates in repairing errors. Now fourth is DNA polymerase beta enzyme. It is member of family of X polymerases. It is mainly found in vertebrates, but few are also present in plants and fungi. And the primary function that it performs is DNA repair. It performs base excision repair. This we will discuss in separate video on DNA repair. Structure. It is smallest and simplest polymerase enzyme in eukaryotes. It is composed of a single protein which is approximately 40 to 48 kilodalton. Now activities. It is not highly active, it is not very processive, and it has no intrinsic exonuclease activity, or it lacks intrinsic exonuclease activity. Therefore, it is involved in mainly DNA repair. Fifth is DNA polymerase gamma enzyme. It is member of family of A polymerases. It is only mitochondrial polymerase. That is, it is only the polymerase which is present in mitochondria, as mitochondria also consist of DNA. Functions and activities. It replicate and repair mitochondrial DNA. Since it is only mitochondrial DNA polymerase enzyme, hence it performs all the function related to DNA replication and repair. It has 3-5- exonuclease activity. Let us see comparisons of DNA polymerases in prokaryotes and eukaryotes. So first of all, comparison of properties of DNA polymerase in prokaryotes. Here we will focus on three polymerases, DNA polymerase 1, 2 and 3. First of all, DNA polymerase 3 are holoenzyme. Holoenzymes are those which are completely functional, and this consists of both enzyme and coenzyme. DNA polymerase 1 and DNA polymerase 2 are not holoenzyme. Now the function. DNA polymerase 1 perform repair and also replication function. These removes and replaces primers. So these fills the gap between Okazaki fragments of lagging strand. Now DNA polymerase 2. These have the repair function and also restarts replication after damaged DNA halt synthesis. We have discussed this in detail in previous videos. You can refer it. So these function as the backup enzyme of DNA polymerase 3. DNA polymerase 3, these are main replicating enzyme. These elongates newly synthesized DNA strands. Now the processivity. DNA polymerase 1 have poor processivity. Similarly, DNA polymerase 2 have poor processivity. These are least efficient. These have the efficiency even less than DNA polymerase 1. And DNA polymerase 3 enzymes are highly processive, or these have high processivity. The 5-3- polymerase activity. Polymerase 1, 2 and 3 all have 5-3- polymerase activity. 3-5- exonuclease activity. All the three enzymes polymerase 1, 2 and 3 has 3-5- exonuclease activity. 5-3- exonuclease activity, only polymerase 1 shows this activity. Polymerase 2 and 3 do not have this activity. Affinity of nucleoside triphosphate precursors. Polymerase 1 has low affinity. Polymerase 2 also has low affinity and polymerase 3 has high affinity. Now the structural genes. For polymerase 1 it is Pol A, for polymerase 2 it is Pol B and for polymerase 3 it is Pol C. Number of molecules per cell. There are 400 polymerase molecules per cell. Number of polymerase 2 molecules per cell is not known and polymerase 3 is 10 to 20 per cell. Nucleotide polymerize at 37 degrees centigrade. Molecules added per minute. By polymerase 1 it is up to 1000. Polymerase 2 adds up to 50 nucleotide molecules per minute and polymerase 3 adds up to 15,000 molecules per minute. Location. All of them, polymerase 1, 2 and 3 are located in cytoplasm of cell. Now the comparison of properties of DNA polymerases in eukaryotes. So it was found that there are around 15 different DNA polymerases present in eukaryotes, but only 5 different types are mainly studied. These are DNA polymerase alpha, delta, epsilon, beta and gamma. So let us compare their properties. First is location. DNA polymerase alpha, delta, epsilon and beta. These are located in nucleus. And DNA polymerase gamma is located in mitochondria. These are involved only in the mitochondrial DNA replication since mitochondria has DNA. Now 3-5- exonuclease activity. DNA polymerase alpha do not have this activity. DNA polymerase delta and epsilon show this activity. Again, DNA polymerase beta do not show this activity and polymerase gamma show this activity. Primase activity, that is synthesis of primer. DNA polymerase alpha shows primase activity. Others, DNA polymerase delta, epsilon, beta and gamma, these do not show the primase activity. Processivity. It is low for DNA polymerase alpha, high for DNA polymerase delta and epsilon. Again, low for polymerase beta and high for polymerase gamma. Fidelity. It is high for DNA polymerase alpha, delta and epsilon, low for DNA polymerase beta and high for DNA polymerase gamma. Now replication process. DNA polymerase alpha delta and epsilon. These are involved in replication of the DNA. DNA polymerase beta is not involved in replication, and DNA polymerase gamma is involved in the replication of mitochondrial DNA. The repair activity, DNA polymerase alpha is not involved in the repair mechanism, and other enzymes, DNA polymerase delta, epsilon, beta and gamma are involved in repair mechanism, or these are involved in DNA repair. Associated with PCNA, PCNA is proliferating cell nuclear antigen, which is responsible for the processivity of the DNA polymerase. DNA polymerase alpha is not associated with PCNA. DNA polymerase delta and epsilon are associated with PCNA because these are major replicative enzymes which have high processivity. DNA polymerase beta and gamma are also not associated with PCNA. Now the comparison of DNA polymerases of prokaryotes and eukaryotes. So in prokaryotes, we have already seen that there are 5 types of DNA polymerase enzymes. DNA polymerase 1, 2, 3, 4 and 5. In eukaryotes, about 15 types of DNA polymerases have been found, but only 5 types are mainly studied. These are polymerase alpha, beta, delta, epsilon and polymerase gamma. Now the functions of polymerases. In case of prokaryotes, DNA polymerase 1 is involved in DNA strand synthesis, proofreading, repair, and it also removes RNA primer, and hence it fills the gap between Okazaki fragments. In case of the eukaryotes, polymerase alpha has both polymerizing activity and primase activity because it can synthesize primer. So it is an unusual enzyme. In prokaryotes, polymerase 2 shows repair activity. In eukaryotes, polymerase beta is also involved in repair of the DNA. If we talk about the main replicative enzyme, in prokaryotes it is polymerase 3 and in eukaryotes it is polymerase delta and polymerase epsilon. Polymerase 4 and polymerase 5 in prokaryotes, these are also involved in repair under unusual condition. Polymerase gamma of eukaryote, it is present in mitochondria and it is involved in the replication and repair of mitochondrial DNA. In prokaryotes, polymerases also show exonuclease activity, but in eukaryotes, all polymerases are not exonucleases. Now the location of the prokaryotic DNA polymerases, these are located in cytoplasm, and in eukaryotes, polymerase gamma is located in mitochondria and other polymerases, that is polymerase alpha, delta, epsilon and beta, these are located in the nucleus. DNA polymerases require primer for the synthesis of the new strand of DNA. In case of prokaryotes, RNA primase synthesizes the RNA primer. In case of the eukaryotes, primase activity is present in DNA polymerase alpha. Hence, it is self-primed. It synthesizes the primer and then small stretch of the DNA, and then it is replaced by the major replicative enzyme. In prokaryotes, both strands are synthesized by the DNA polymerase 3. Here, the core polymerase are present in the form of the dimer, and each of them synthesizes one strand. In case of the eukaryotes, DNA polymerase delta and DNA polymerase epsilon, which are the major replicative enzymes, synthesize opposite strands. If one strand is synthesized by DNA polymerase delta, then the other strand is synthesized by the DNA polymerase epsilon. In case of the prokaryotes, no polymerase switching takes place because here the primer is synthesized by RNA primase. In case of the eukaryotes, polymerase switching occurs. First of all, DNA polymerase alpha synthesizes the primer and then small stretch of the DNA. And then it is replaced by the major replicative enzyme DNA polymerase delta or DNA polymerase epsilon. This is known as polymerase switching. We know that DNA polymerase 3 of prokaryotes has high processivity, and this is due to the beta sliding clamp. In case of DNA polymerase delta and epsilon, their processivity is due to the association with pCNA, which is known as proliferating cell nuclear antigen. This is all for today's video. In the next video, we will discuss about DNA replication in prokaryotes in detail. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.